Let's look at these stabilizing forces one by one. First of all, hydrophobic interactions occur in between nonpolar alkyl group and the aromatic groups. Because they are repelled by water molecules, the hydrophobic amino acids are always buried in the protein core. In the case of hydrophilic interactions, you have the attractions in between the polar or the ionized side chains or the R groups. They can also interact with the water molecules on the surface of the protein. Then you have salt bridges. So these are the ionic interaction of charged R groups. And of course, you have hydrogen bonds and the disulfide bonds that you have already studied before. Here I just want to show you how strong are these chemical interactions. The strongest bond is of course the covalent bond, which is found inside the peptide bonds within the amino acid backbones. It has a free energy level of 356 kJ per mole, which means you will need this amount of energy to break just 1 mole of chemical bonds. Coming next is disulfide bond, which is another type of covalent bond. So it is still very strong because it has about half the strength of a conventional carbon-to-carbon covalent bond. Coming next is the salt bridge, which is almost 10 times weaker than the disulfide bond. And then you have the hydrogen bond, which is the weakest chemical bond in all these interactions. So it has a free energy level of about 2 to 6 kJ per mole. But bear in mind, every amino acid is capable of forming the hydrogen bond with one another. So collectively, they can form a considerable amount of force. So another type of interaction is called the long-range electrostatic interaction. So which is similar to salt bridges, but they happen at a larger distance. So the bond strength is variable. Last but not least, you have the van-der-Waals interaction. So you always thought that it is the weakest of all, but it is actually pretty close to the hydrogen bond. And it can be even stronger in the protein interior. Okay, given the knowledge of chemical interactions in proteins, can you predict which interactions will break first when the protein is heated? So here is just a suggested answer purely based on logic. Well, first of all, the hydrogen bond is going to break first because it is the weakest interaction of all, as it has the lowest free energy. It is followed by the hydrophobic van-der-Waals forces, then the hydrophilic interaction between the polar side chains. After that, it will be the salt bridges, which involve charge groups, followed by the disulfide bond, and eventually it will be the peptide bond, which is a covalent bond. So this unfolding of protein is also known as protein denaturation.